Our final presenter is Lauren Kuehne, who's going to talk about lessons learned from 20 years of knotweed management in an unregulated river. Lauren is an environmental scientist who focuses on supporting management of aquatic and riparian systems. She received her MS in Aquatic and Fishery Science from the University of Washington in 2012, where she was a research scientist until 2019. Her work encompasses diverse conservation issues, including fish, forests, invasive species, and noise pollution. Most of Ms. Kuehne's research is in Western Washington and collaboration with agencies, tribes, and nonprofits doing on-the-ground restoration. Her research also reflects a commitment to science communication and outreach, including community science, blogging, public talks, media presence, and op-ed articles.
Okay, hopefully everyone can hear me. Okay, I'm traveling today and my flight got delayed, in fact, at the airport. And hopefully the announcements won't be too bad. But I'm going to, thanks for everyone for sticking in today to listen to this talk. And I'm excited to share work with you. This data set that I'm going to be talking about, one of the data sets I'm going to be talking about, has been 22 decades in the making. And so I'm excited to share that. And then I'm actually, this is kind of two talks about 2/3 of the way. I'm going to switch gears and talk about an herbicide trial done in conjunction with Dave Heimer at the state of Washington. But first, I'm going to talk about lessons learned from 20 years of management. And unfortunately, because I was on an airplane, I missed Bank's talk. Hopefully we're not overlapping too much, but maybe we will be confirming what the others said. So the data set that I'm going to be talking about today is out on the Hoh River on the Olympic Peninsula in Western Washington. And 10,000 Years Institute has been the lead entity now. 10,000 Years leading this work, but it has been incredibly collaborative. It started by Hoh Tribe, has been sustained by multiple tribes are in that area, U.S. Forest Service, state agencies have worked on this effort for over 20 years. 10,000 Years Institute, what they do though is they work on best practices for integrated restoration, and they focus a lot on riparian ecosystems and invasive species management and removal. And they really, 10,000 Years Institute really likes to integrate science into their work. And so that's part of the reason why they've been documenting this data for a long time while they have done restoration. So Hoh River is very famous. It's the site of the Hoh rainforest, right? And it's one of the several, so many of the rivers, most of the rivers out on the Olympic Peninsula are a little unusual or unregulated. Most of them are undammed. And so the Hoh River is like this 80 kilometers of undammed river on the Olympic Peninsula. It is a rainforest, so it meets the criteria for a rainforest. Originates at the Hoh Glacier on Mount Olympus, and of course the Hoh rainforest is world famous. It's actually a World Heritage Area in the national park. It has a lot of abundant wild fish population because it has pristine, relatively pristine habitat for salmon and intact riparian areas and habitat. So we have a lot of fish populations out there, and fishing is obviously of enormous cultural importance to the tribes there, as well as recreational fishing too that for that area. Now, the history of this invasion, the opportunity, if you will, to study this happened because, well, I hate to call it an opportunity, but this is what happened. In 1999, a river channel avulsion captured a knotweed clump and it moved it into the river, probably from a kind of a farm garden. So that river channel avulsion happened at basically river mile 30. And since then, it was detected a couple of years later, but by that time the knotweed had spread. It was detected pretty shortly after this storm event, or this river migration event that avulsed into the knotweed into the river. And so between 1999, it was detected in 2001, so about 2 years of some survey and some treatment before treatment was actually started. And the first funding was in 2001 from the Bureau of Indian Affairs. And that's actually, this is a picture of Jill Silver, 10,000 Years Institute flagging knotweed on the Hoh River. So 2001 was a long time ago. the interesting part about this story is that Jill and the Hoh Tribe and other entities, the Wild Fish Conservancy, and I mean, there's just so many. The acknowledgement side is hugely full. Have for this 20-year period, we started with knotweed in 2001, and then did survey and treatment and control work through 2010. Well, actually, through all of these. But in 2010, started adding treatment and control for other invasive species, so Scotch broom and reed canary grass, and so this effort has been sustained over this entire time period. You can see, you know, diverse forms of funding have been tapped, diverse initiatives have been utilized to make this a sustained. Sorry, I'm going to let that announcement go a little bit while I change slides. Thank you. And like I said, it's, you know, 10,000 years is kind of the lead entity, if you will, but it has been an immensely, there have been so many collaborators, so many people have done the survey work over this period of time. Here's a picture of Jill giving a training to folks. Now, this is a beautiful photo. It's nice to just capture the wild and relatively pristine area, but it also captures the magnitude of the challenge here. This is it's an unregulated river. It's very dynamic. It moves a lot. There's a lot of sediment that moves around. The gravel bars move. There's a lot of vegetation and a lot of survey to cover. So many of the challenges over the years have been, how do we find these plants in this dynamic, not only large area, but a dynamic area from year to year. So survey challenges include the diversity of habitats. Knotweed will establish on gravel bars. It will establish in willow thickets. It will establish in forested areas. It will establish in these, you know, log jams, all of which have enormous, the Hoh River has enormous areas, all of these types of habitat. And so finding ways over the years to comprehensively find the knotweed as this has moved has been an ongoing challenge. And so, for example, there's the needle in the haystack, if you will, of the challenge of what trying to remove knotweed from this river has looked like over time. Just to give you a summary of, you know, what that overarching effort has looked like, the monitoring and restoration work in general. Of course, it's changed from year to year. It has had to be flexible and change from year to year, depending on funding or, you know, crew logistics or what kind of personnel were available, these kind of things. But overall, the control area is downstream of that introduction point at River Mile 30. It has continued almost every year since then. So in most patches and in most years, knotweed has been surveyed and treated in the earlier years with glyphosate injection, and then they switched to foliar species. And then starting around 2008, switched to incorporating imazapyr. And for the most part now it's imazapyr that's used almost exclusively now. But overall, for the most part, it's been consistently surveyed and treated in each year in most reaches. This would be our monitoring and control area. You know, this is the area that gets of monitoring and control from every year from 2002 to the present. So when Jill and I have been talking for many years about doing something with these data, because every year that they surveyed and controlled, they also kept very careful records of what they treated and where they surveyed and where they found knotweed. And so all this data was being collected. And so what we saw as an opportunity, well, let's try and do something with this data to inform management in other rivers. And there were a couple different stories to come out of that. One is that because we are analyzing knotweed presence and establishment over 30 river miles, 90 kilometers, we thought that we could establish, say, well, what are these landscape factors that, you know, where do we see knotweed showing up again and again, no matter how much you treat it? And where are we seeing it persist and where are we seeing it establish? You know, where is it piling up on the river? Where are we able to eradicate it? And so we thought that it might be possible to identify the landscape factors that would allow us to assess where these hotspots are and if there are, you know, factors associated with persistence. But the other part of it is that every year there is information that's been collected in terms of anecdotal observation, but you know, they may be anecdotal, they may be observational data, but if you observe them over 20 years, they start to pile up. And so we wanted to try and find out, there's also, you know, within these 20 years of reports and documentation, there's lessons for adaptive management because of course, 20 years ago, we knew less about knotweed than we do now. And so there's a story in all of these documentation around how do we adaptively manage and learn as we go. So that was our two research goals. Was one, to analyze this 20-year data set, and the second, we'll try and capture, what are the lessons that we can take, and then to help people that are dealing with knotweed in other river systems, or to communicate the management story. So the first one I'm going to talk about is modeling. I like nerdy modeling stuff. Hopefully this doesn't bore everyone, but we wanted to say, well, what are the landscape factors where we're seeing, associated with establishment persistence of knotweed? And so what we did is we have these knotweed observations, right, in our monitoring and control area. So here, this is knotweed points from 2002. Now we have data points like this, spatially referenced data points like this from 2002 to the present, to 2023 each year for knotweed for most of the river reaches. So then what we wanted to do is we wanted to say, okay, well, we've got this longitudinal record. And we had some hypotheses that we said, okay, well, we think that, proximity to the source will have a positive effect. The closer we are to the source, the more knotweed we'd expect to find. The treatment year, obviously we're treating it every year, so that the knotweed should go down every year. The active channel width, we thought that a wider channel should be associated with more knotweed, channel braiding, positive, channel sinuosity, right? All of these factors, we had these hypotheses about the direction that it would be pushing presence and abundance and density of knotweed. Down here, just to show you this percent forested, that means the percent forested in, at the edge of the channel, right? And the percent emergent wetland is also at the edge of the channel. So that was how we set up, how we wanted to go about doing this modeling to assess these factors. The steps to do that, so we have our knotweed points, and we're going to assign them to these river kilometer bins. So here is 30 river miles, and each of these is a river, the center of the river kilometer. So this becomes our river kilometer bins. And we're going to create covariates for each one of those points. Here's our monitoring and control area. Here's again, like our 2002 knotweed points. And then we assign these knotweed points to its river kilometer bin, and then we create covariates, and then we're going to model those with the years. For people that want to know how we came up with these covariates, here's the mouth of the river right here. And so each of these, this is like quarter mile points. And so to do the active channel braiding, we take 4 samples per river kilometer, and you just measure the active width and you measure the amount of braiding in it. For the percent forest, we did that by taking each of those points again, and then taking this 500-meter buffer around these single points. And we're binning it by river kilometer and then creating these covariates. And then we're going to model that for folks that like modeling, to know about modeling. We're creating these models out of our variables here. And then we use model selection using AIC. And then the top model, we're going to evaluate the effect sizes. The way that we did this is we created a bunch of different candidate models, and then we used AIC to say, well, which one of these models is our best fit? And then that final model has effect sizes. For example, we'll say the effect size of channel braiding is this, and we'll look at those results in just a minute. Okay, this is the first result slide. It was the best way that we could figure out how to create these data or show these data, but it's not exactly intuitive. So first thing that you're looking at is year. Okay, so year, we're starting down here at 2002, going up to 2023, right? So time is moving up through the top. And then on this bottom, we have river kilometers. So this is the mouth of the river. And then this is our most upstream extent, right? And sorry, the green, the density is the density of the number of knotweed observations in that year. And so the darkest green is going to have a lot of knot observations, whereas white is basically 0 knotweed observations by river kilometer. So this is 2002, and this is the knot points in that year. And so what we're seeing here is sort of a correspondence between the knotweed densities, right? And in that year, the gray means that it was not surveyed. So now that we've sort of oriented ourselves to this plot, the take-home message on this is that we see pretty effectively through time the reduction in knotweed density. Obviously, the highest density was up here near the source, early years, 2002 to say 2010. but then we start lots and lots more white, where we're having basically zero knotweed. And then that has continued, as we come through time, we're seeing less and less knotweed in each of these river clumps, and that's because of this sustained survey and control. So then here, what I'm going to show you is this is actually the results of the modeling. So we had those hypotheses about these different factors, right? We said the proximity to the source, and we said year, and we said, well, what would these have positive or negative effects? And right here, what I've put is a check mark if it lined up with our hypothesis. So for example, proximity to the source was positive, more knotweed closer to the source. Year, was negative. Here we see the negative effect of year. Sorry, my mouse, I keep trying to, there it is. The one that didn't line up, so sinuosity, so a more sinuous channel, we see higher persistence or establishment of knotweed there. Channel braiding did not. So more braided channels were not, they were actually associated with a negative. And so there's more to explore there. Percent forested was a little bit negative. And then here, this was a bit, well, it wasn't surprising, but the percent emergent wetland that was in, these are 500-meter buffers. So percent emergent wetland had a strong positive association with knotweed presence. So I think this was really, it's interesting. And it's good. It shows us that the landscape factors can predict. We can, at least take an initial cut at saying, well, what landscape factors where we might expect to see more knotweed and where we might see it being harder to eradicate. However, what there is to work on here and refine this is that the total variance that this model is explaining, I mean, it depends. I showed this to someone else and they were like, this is explaining a lot of variance. I think it can do better, but there's a lot more that we can do in terms of adding in new variables or modifying variables. So then the second part of this was, how do we summarize these adaptive management lessons? So I mentioned that in each year, there are these documentation, and mostly this is in the reports. But what we wanted to do is try and capture and summarize these persistent, consistent observations and consistent trends. And so I'm not going to go through each one of these, but we're going to be we're preparing a publication, and I can share this information with anyone. What I'm going to show this slide is just a summary slide. So from each of these, 20 plus years of observational data, what we have is that from year to year, the annual control rates are quite high. We're seeing good treatment and control of individual plants of knotweed. And then, of course, what I showed you in that earlier slide is that we're seeing the knotweed bias. We're seeing the number of stems go down. We're seeing the clusters go down. We're seeing the height of the plants going down. What I didn't show you, and I have data on this as well, but what we were seeing at the same time is that in years with large flooding events, we were seeing lateral and upland spread of knotweed into the upland terraces and then pushing out into the river. So the challenge, of course, is that our survey and control effort, even though the knotweed is going down, you have to look harder for it. It's smaller plants that are more spread out, and they're pushing associated with flood events, they're pushing into these new areas. Basically further out from the river. So it's not a grim story. I don't want to call it a grim story. This is the story that Jill and I are grappling to tell, because this is the reality of managing this plant over a long period of time in a river. So some takeaways and next steps. We did show that Jill and her consortium, I guess, of folks, they have showed that they can reduce knotweed through time, even in a changeable dynamic river, which is really quite astonishing and impressive. We can use landscape factors, to explain areas of establishment and persistence. And then I think that, one thing that is really important to me as a scientist is that, I think that it's difficult to document your monitoring and your control and your survey efforts. However, if they can be sustained over time, it can help us generalize these lessons for other areas. So some next steps, to keep surveying these small populations. And also I think what Jill has found is that because there's less knotweed to survey for, she has been able to use crew time to survey and manage it for these other species. So that has proved to be a useful kind of way to extend the work and keep it sustaining. The next step, we want to refine this landscape analysis using more factors and higher resolution. And then, I mean, we could, once we have this model, we could use it to predict invasion risk and spread and persistence on other rivers. And then lastly, I'm always doing this in general, but I think hopefully this work supports, development of data collection and data repositories and data information sharing at different scales for invasive species. So that is kind of an analysis of like this landscape scale, long-term data. I have another project that I've worked on, and this is in conjunction with Dave Heimer at DFW, where we looked at sequential use of Diquat for managing invasive knotweed. This is a pretty straightforward herbicide test. However, there's a lot of elements to it that I think are really important with respect to knotweed that I'll touch on. The reason that Dave initially sort of approached me, or we talked together about doing this, is that he works with tons of folks in the state that are trying and grappling to manage knotweed on these different areas. And this is just sort of from an informal survey that he did from folks that manage knotweed. Their treatment window is July to October. For the most part, it's not recommended to mechanically treat. Most people are using imazapyr, foliar. For the most part, you can get one site feed season. First year efficacy is pretty good, but successful control, of course, requires multiple visits. And then access is always a challenge. Land ownership, other, getting to riparian areas are always challenging. The idea is that we need to keep evaluating, and what Dave has mentioned is, and for me too, looking back at the history of Spartina. You look back at those older reports, in the early days where they were really grappling with how to do this, and they would try something and say, okay, well, that didn't. So I think that there's a need for this sort of ongoing building of the toolkit. On that note, what we were motivated for is to look at Diquat. Now, Diquat is a contact herbicide. And the motivation was partly because Diquat is labeled for aquatics. And Dave in particular was interested in seeing it be added to the aquatic noxious weed NPDES permit. However, there is also other work that has suggested that instead of mechanically mowing knotweed, seems to just make it mad. Different studies have tried to use contact herbicides as a kind of a chemical in order to improve control outcomes. There's two reasons for this. For one thing, it just cuts the biomass back so that you can get to it. The second reason, though, is that by forcing the plant to recover within the growing season, there's a hypothesis that it improves the uptake of a subsequent treatment with imazapyr glyphosate, a systemic. So Diquat kind of met all of these things. It would allow us to test this contact herbicide. It is labeled for aquatics. However, we didn't have any label rates for invasive for how to treat knotweed. So we saw an opportunity to kind of fill in a few different gaps. And this is a chart of all the different studies for knotweed and the different herbicides that have been tested. And if there's a gray box there, then it means that it has been tested, and a star means that it was considered very effective. And when we looked at how often Diquat had been tested on a knotweed, it was basically completely non-existent. So we thought, well, worst case scenario, we see and test this out and see what it does on this plant. It also has a different mode of action. So this is, what we did. We had a pretty straightforward experimental design in our timeline in early 2021. We set up our plots. We did an early season Diquat treatment, and then we evaluated at 2 weeks after, at 5 weeks after, at 8 weeks after. And then we did that sort of standard imazapyr treatment. And then we evaluated 1 year after. We did this in 3 sites and 4 replicates in each site. And then we had 3 Diquat concentrations, low, here, medium, and then high. And I can't remember exactly, but I believe it was based off of cattail. The concentration that Dave has seen with different cattail. That was kind of where those concentrations came from. And this is an example of a block. So it would be 4 treatment plots side by side. And, I just kind of threw, you'd have one of each of these in each treatment block. And then in each of our sites, we would have had 4 of these replicate blocks. Now our plot, this is going to be a little bit later. This is how it was challenging because we knew that the Diquat was going to reduce the biomass. So how do you assess effectiveness when you know that you're going in and you're already cutting, you're doing this chemical mow, if you will. So what we did is we have these plots that are 6 by 6, and we had 2 1-meter quadrats in there. And we had to measure multiple responses. We measured percent cover. We measured percent injury. However, the plants recovered really quickly. So percent injury kind of fell out. It ended up, but we also did stem counts in each of the quadrats, and we measured stem heights. And both of those became important in being able to assess a change in what we saw. Okay, this is another, I only have one plot because I really, I know that my time is getting short. So what I'm going to show you is the results for the stem heights. And what we did from stem heights is we extrapolated biomass. So this pre section, this is before we treated anything. And then over here, each of these lines is low, the pinks are low, medium, and high, and then our black dotted line is controls. And over here, this is stem heights. So what you want to notice is that, pre-treatment, all of the plots, the stem heights, the distribution of stem heights looks exactly the same. When we treat with Diquat, and then 8 weeks later, all of our Diquat treatments are responding very similarly. And basically what you read this as is there's fewer, so this is a higher number of larger stems, this black line here in the control plots, and all of them have far fewer tall stems. Now, a year after, we've gone in at this point and we've treated with imazapyr, and so one with Diquat and one with imazapyr, and we've gone and looked at it a year later. A year later, we're seeing a difference. The low Diquat treatment is looking far more like the control plots. But the high and the medium Diquat treated plots do look different than the control plots. And so what we think is that high and medium Diquat concentrations are somehow, we're able to make the imazapyr more effective. However, we needed to do a longer term trial in order to really assess that. But basically the take homes from this is that, at 8 weeks after treatment, all of the Diquat treated plots are substantially reduced. So the mass is not fully recovered. 52 weeks after, a year after the combined treatment, the low one looked really similar to the controls, but the medium and high concentration were reduced compared to controls. And we were able to use stem counts and heights to estimate biomass. I think that that's really important because, you can have a difference in stem counts, or you can have a difference in stem heights. So the way that this looks in the field, this is our control plot, before any treatment, and this is our control plot 8 weeks after, and this would have been our high Diquat treatment before, and then basically they kind of look the same. So, the plants recover by 5 weeks after treatment, but the biomass is still significantly less at 8 weeks after treatment. And so the take home is that Diquat is going to reduce your biomass, but not your stem counts. And then we think this facilitates the subsequent imazapyr treatments. The Diquat and the imazapyr are at least as effective as the imazapyr alone, but we need to do a return trial to see if it actually improves the uptake and a longer term treatment. This method that I highly recommend the method of evaluation that we used, and I'm happy to share it with anyone. We have a published page that can share it. But I think that it's really robust, and it's actually not that, it's not that difficult to collect these data, and it allows you to assess the difference between stem count and biomass. We obviously by treating with Diquat, we're reducing the risk of flowering. And then it reduces the knotweed biomass for easier application and less product in the imazapyr and glyphosate. And we think that, when folks are struggling, they have logistics, they've got crew, they've got access issues. Using these extended toolkits can sort of help us extend our treatment windows and have alternative options sometimes in order to treat plants. And then we provided information to ecology to support inclusion. This is trying to tie the two things together, is that, I mentioned at the beginning, I am an environmentalist. I try and do research that promotes management and conservation. What I find is that I think that there is a need to do research at different ways. We need to understand how to have more effective treatment of, say, knotweed. We need to understand how to get that herbicide into the rhizomes, how to get translocation better. Have chia pet, types of syndromes to deal with. And then we have things like hybridization, all the way up to the influence of floods and how things like change and, glacial retreat are even going to impact. And then, of course, the overarching trend in warming habits. So I think that we have a need to walk and chew gum at the same time, and to do research that addresses all of these, and that we find ways to integrate this. I think that's all that I'm going to go ahead and stop sharing. Have you observed knotweed cuttings made by beaver on shorelines? We observed hundreds of cutting made by beaver germinating on the shore. East Fork Lewis County collection was key to mitigating spread. So I've seen that on the Chehalis. Actually, I was out with Elizabeth Seebacher and we were watching Nutria, I think, literally carrying chunks of knotweed across in its mouth. And so at least anecdotally, I would say that that seems to be a thing. I don't know if there's a lot of beaver in the Hoh. There's a ton of beaver in the Chehalis. And so that would be a place to kind of think about. But personally, I've seen nutria specifically swimming around with knotweed chunks in their mouth, and we've seen beaver chewed cuttings of knotweed as well in the Chehalis. And then someone said, on the Hoh River, did your team switch to imazapyr to reduce knotweed resistance to glyphosate, or because it was more effective? That was in those 20 years of adaptive management lessons that I've now forgotten. What was the reason for the switch to imazapyr? I think it was, they started out by incorporating, they started out by adding imazapyr to the glyphosate, and noticed that it was more effective, and then ended up switching to imazapyr. But there were concerns because of residual. They were finding that the imazapyr resulting in killing off of native vegetation, thinking probably because it maybe has a higher residual. So they're still using imazapyr, but I think the goal was to try and be more targeted and try and avoid using it with native or around the vegetation. And so, yes, the residual, if you email me, I can send you the summary that talks about the switch to imazapyr, and yes, there was some issues with seeing it being impacting native vegetation. I have been apprehensive to use imazapyr, so okay, 20 kill plants within a 20-foot reach. So that is another good question. Yes, similarly, again, in these 20 years of documents from the Hoh River, that it seemed to be an issue. And I can actually, again, I'll put my email in, and I probably can get an updated answer to that question from Jill. On how the imazapyr has been going, because when they first started using it, it was an issue. Does Diquat have a similar result? I don't think we know. I mean, in our treatment plots, for the most part, it was 100% imazapyr. I'm sorry, 100% knotweed in our treatment plots. There weren't other vegetation. So I'd have to look up the selectivity of Diquat. Possibility of biological control agents for knotweed. I don't know I know that Jen Parsons would be the person to talk to about biological control. And I believe that there are some not biological control agents in the works. But I don't know which ones they are. But Jen Parsons is on top of all the biological control stuff in this region. And then, did you see any biomass difference with the Diquat used in forested areas versus open, full sun areas? That is an excellent question. So not with the Diquat. In fact, we had 3 sites that varied. Sorry, I'm trying to return back. Our 3 sites varied quite a bit in the amount of forestation. And we didn't really see a difference in change. What we did see was that the knotweed overall, the biomass of the knotweed in the forested sites was not as robust, as tall as it in the forested areas as it was in the sun areas. So it obviously does vary. We get lots of plenty of density of biomass in these forest areas. But the height of it didn't seem as large, and we just weren't getting overall amount of biomass to start with. So hopefully that answers your question and maybe lines up. And I think that lines up with what they're finding, what Jill has found on Hoh, is that it establishes and does plenty well in forested areas. It just probably isn't going to reach the same heights or maybe the same density, but it doesn't stop it from establishing and growing and persisting. So